Ubiquitin is a small, highly conserved, heat-stable protein present in all eukaryotes. It is the most important regulator of protein homeostasis. It has 8 covalent, 3 main hydrophobic ubiquitination patches, as well as a very flexible C-terminus where the substrate is attached by an isopeptide bond. The predominant function is marking other proteins, thereby targeting them for transport to the proteasome for degradation. Also has roles in gene expression, mitosis, cancer development, and cell cycle processes. When binding, ubiquitin adopts a pincer mode where its motional dynamics strongly affect its binding affinity. This is a combination of conformational selection and induced fit, which greatly lowers entropic barriers. High plasticity and therefore high affinity are motifs, but not so high as to be promiscuous. The pincer mode directly influences the geometry of the 3 hydrophobic patches at leucine 8, isoleucine 44, and valine 70. Binding partners form complexes according to the coordinates set by the geometry of these patches under pincer conformations. In an experiment featured in the Journal of Molecular Biology, serine mutations were induced on leucine 67 and leucine 69, adjacent to the 6 amino acid C-terminus end. These mutations have almost no effect on the overall structure of the protein, but they reduce the rigidity and cause a slight displacement of the C-terminal end. The hydrophobic patches and the structure remains completely intact, but the mutations slow down the timescale dynamics enough to cripple its affinity. Movement is so important that the slight alteration in timing cripples ubiquitin's ability to function.
While monoubiquitination is possible, ubiquitin often acts as a polymer, forming a multitude of different chains. These chains are differentiated by which lysine residue they are linked to, with different types of lysine linkages having different properties and conformations. A lysine 48 linkage forms a tight, discrete cluster, which often marks for proteasome degradation. The interactions to the isoleucine beta sheet, which is buried when not interacting, are exposed by conformational changes when it's bonding. This results in the open and closed form as discussed in the pincer. Lysine 63 has much more movement and freedom than lysine 48 change due to the lack of strong covalent interactions. This leads to many more conformations and much more extensive and varied structures. Lysine 63 linked chains are associated with exocytosis, DNA repair, and translation. Here you can see a very visual representation of the different types of change, with K63 linked chains looking more like a beads of a string type model, and the lysine 48 chains looking more discrete and compact.
Okay, here you can see some images rendered in 3D by Swiss PBD viewer. Here we see the ubiquitin molecule. The C-terminal end is colored in green, while the 3 hydrophobic patches of relevance are covered in red, blue, and purple respectively. The hydrophobic patches are very essential in the protein-protein interface between 2 ubiquitin molecules. Here we see the 2 lysines that are essential in linkage between ubiquitins. Cyan represents 63, the more fluid form of linkage, and 48 is orange, more rigid, tightly packed chain. And that concludes our presentation. Thank you for listening.